Okay, so the first one, because last week you learned about the different types of primary tissue. There are 4 types, if you still remember. And today, it's just a brief overview on different slides. Just I want to show you the atlas of the histological stain. Basically, the H&E stain micrograph of different types of tissue that made up the organ system. So learning outcome is just to understand the microscopic structure of tissue organization. It's just not to understand, but maybe you can have a brief overview of different types of cells or tissue that made up our organ system. And you are able to identify and describe the major cells, the primary tissue that comprises the important organ in our body. Okay, so the lecture content, I pick not all, but several important organ system that we were going to look at. The first one is the circulatory system. Basically, the structure, it has the inner lining and intermediate muscular layer and also the outer connective tissue. So basically, our heart, main organ in circulatory system, is made up by epithelial cells, the inner lining, and it has also different types of muscular layer and different types of connective tissue that are lining the outer layer. So I give you example of the myocardium, the wall of the heart. So this one is the longitudinal section. So it have the cardiac muscle fiber, and also you learned last week it has the intercalated disc connecting neighboring cardiac cells. And also it has the central nuclei, and the cytoplasmic is in a form of cross striation. But if you look at in the transverse section, it have extensive capillary here. So the transverse is when you cut like this. So you'll see the extensive capillary network system, the C, in the myocardial fiber. So the myocardium is very high level of vascularity. So for the heart valve, so this is a micrograph of the valve. So the valve is the fibroelastic tissue covered by the endocardium, and there is a dense central plate of collagen lamina fibrosa, the LF here. So you'll see this picture last week, but I didn't show it very clearly. So this one is much better micrograph. Okay, there's a scattered elastic fiber, which is in black stain. Okay. And next is the skin. So skin, there are 3 layers of skins: the epidermis, the dermis, and the subcutis or the hypodermis. Okay, so this is the skin architecture. So the top one, the outer part, is we call it as the epidermis. It is a surface layer of stratified squamous epithelium with the surface protein of creatine K. So it is a keratinized protein. So for dermis, it is adjacent to the epidermis. So the PD is actually the first layer of the epidermis. So the RD is the second layer of dermis architecture. Okay, so the PD, it contains the fine collagen fibers and a numerous blood vessel. And the RD is actually arranged in collagen and also elastin fiber. You can see here, there's a whitish color, irregular shape of the collagen's accumulation. And for the subcutis or hypodermis, a layer of adipose tissue here extending downwards together with the connective tissue. And it also contains the EG is actually the sweat gland that secrete your sweat, and also apocrine gland. Okay, so as long as you know there are different layer of skin, then it's already enough. Okay, next, in the skeletal system. So skeletal system comprises many types of supporting or connective tissue. So we have bone, cartilage, joints, ligaments, tendons, right? So this is example, micrograph example of hyaline cartilage, which is the most common type of cartilage. So it forms a precursor of bone in the developing skeleton. And mature hyaline cartilage is characterized by aggregates of the chondrocytes embedded in the matrix. Okay, so the outer part is actually here, the perichondrium, the P, composed of parallel collagen fibers here. And also the CB, the fibrocytes, which later will transform into the small chondroblasts. Here, as you can see. And the chondrocytes of the formed cartilage is usually formed in a cluster. So at the upper layer, it will become the chondroblast. But for the chondrocytes, it usually extending downwards in a form of cluster, usually 2 or 4 types of cell, 1, 2, 3, 4, in a form of cluster here. This is also a cluster of chondrocytes. And the pinkish staining is actually the cartilage matrix. And also the compact bone. So this is a micrograph of H&E stain section of the calcified bone, the smooth bone. Okay, so the outer surface is periosteum, and the OCL is actually the outer circumferential lamellae, which lie between the periosteum and the Haversian canal. So this is another example. So T, trabecular bone, and FM is actually the marrow, the bone marrow space filled with the adipose tissue, and AC is the cartilage neighboring the trabecular bone and also the cortical of the bone. Okay, so there is a connection, different types of tissue that made the bone system. Okay, so for immune system, the major organ that involve immune system is actually the lymphoid system, comprises of bone marrow, thymus, lymph nodes, and also spleen. And also there are different types of cells that involve for cell-cells interaction in immune system. So this is an example of the spleen. So it have consists of the white pulp and also the red pulp. So white pulp consists of the lymphoid aggregation, and red pulp making the bulk of the whole organ. And also spleen has fibroelastic capsule here, which cover the mesothelium, the organ. Okay, so this is a part of the human spleen. So this is the palatine tonsil. It organizes masses of lymphoid tissue which along the lingual, pharyngeal, tubal tonsil form all the ring. So the luminal surface is covered by stratified squamous epithelium, the E. Here that deeply invaginate the tonsil, forming the crypts. Yeah. So the surface layer is lining by stratified squamous cells. And also it have the dense collagenous hemicapsule here. So also collagen tissue also present in here. Okay, and F is actually the numerous lymphoid follicle that presents in the tonsil. And next is the respiratory system. So there are 4 main histological layers, which is the respiratory mucosa, submucosa, cartilage and muscle layer, and also adventitia is actually the other structure, the adventitia. So this is a part of the nasal mucosa. So for the mucosa of the nasal cavity consists or lining of the pseudostratified ciliated columnar epithelium. So if you can see, this is actually the pseudostratified ciliated. You can see the cilia at the outer part here. Okay, so it is supported by lamina propria, which is rich in blood vessel, and it also have a serous and also mucus gland, okay, to secrete the mucus. And this is part of the trachea. So the inner layer of the trachea wall, and this is the respiratory epithelium. You can see there's some ciliated structure here, and lamina propria form a band of fibroelastic tissue, and also present the submucosa that contain numerous mix of seromucinous gland to produce the mucus. Okay, so mucus will help to trap any pathogens so that it will be flushed away from the trachea part by coughing or sneezing, okay, something like that. So that is the function of the mucus. So and also it have the dense fibroelastic tissue extending downwards. And this is a primary bronchus, part of primary bronchus. So the micrograph showing the respiratory epithelium, which has some goblet cells, and also the upper lamina propria contain more elastin fiber. And the M is actually a layer of smooth muscle cells, which later becomes more prominent in more distal bronchi. And submucosa contain fewest here, whereas G here, seromucinous gland, also to produce the mucus, and C is actually the cartilage support. Okay, this is a micrograph of the bronchiole. Bronchiole also part of the lung structure. So its wall composed of cartilage or submucosal gland and smooth muscle cells here, and distant thin wall of pulmonary artery branch, which is the art. So this is only one bronchiole. This is the type of abnormal cells. We learned about the abnormal cells last week, how it form, what triggers them. But this is the actual micrograph of lung cancer cytology FNA sample by gene sustaining. So this is the normal epithelial cells, but this is adenocarcinoma cells. So the normal epithelial with the exhibit expected the columnar shape. And also if we zoom it with higher magnification, we can see cilia that present on the luminal surface of the normal cell. Okay. But for the malignant cells, forming a mass of tissue with prominent nuclei. If you can see, there's a prominent nuclei here, much bigger compared to the normal cells. Okay. And also scanty cytoplasm. So because the nucleus bigger, so it occupy most of the spaces of cytoplasm, make it cytoplasm very small in volume. And next, gastrointestinal system. So the functional layer involve mucosa, submucosa, muscularis propria, and also adventitia. So for example, this is the H&E, all of these are H&E staining of squamous mucosa, gastric type secretory mucosa, intestinal type absorptive mucosa, and colorectal type absorptive or protective mucosa. So because our gastrointestinal tract is larger and big, right? So this is a part of the staining showing different parts of GI tract. Okay, so let's look at the micrograph of the esophagus, the lower third of esophagus. So the lumen of the esophagus is lined by thick protective stratified squamous epithelium here. If we look at in more higher magnification, we will see it is actually stratified squamous epithelium. And underlying lamina propria is quite narrow and contains scattered lymphoid aggregate here. And also MM is muscularis mucosae, presence neighboring to stratified squamous epithelium. Okay, and the muscularis propria is thick and inner circular have the CM, the inner circular, and also outer longitudinal layer. So that is the lining. So this is could be the transverse section of esophagus, the lower third of esophagus. And this is the structure of duodenum, part of duodenum. So the micrograph showing the mucosa has villus form present here. And there is different types of glands, Brana's gland, that mainly located in submucosa. Okay, and there is also mucosa villi here, lining the outer layer. Extending downward, there is two different types of layer, which is the circular layer and also the longitudinal layer. Okay, so this is a colon, H&E staining, and Alcian blue or Van Giesen, both are Alcian blue or Van Giesen, and also the H&E staining. So this is a very clear of structure of colon. Okay, so this is the inner circular layer, and this is the longitudinal layer. Just want to show you different parts of the colon. And this is micrograph of colonic adenocarcinoma, abnormal cell formation from the colon, and malignant epithelial cells form disorganized abnormal glands that invaded into the adjacent tissue, and the malignant gland of the tumor have invaded into muscularis propria. So if you look at this picture, you can see there is a disorganized of the cells organization, and there is small bundles of muscle, the MU, so can be identified. And there is also area of necrosis, which is dead tissue. And also colonic adenoma, a benign but precancerous tumor of the colon. So if prolonged, then it can be a malignant tissue, then it can transform into malignant type. So epithelial cells seen have larger nuclei, and there are obvious mitotic figure. It could have many nuclei present in one types of cells. And the next one is liver and pancreas. So this is a micrograph of a solid organ of the liver, which composed of tightly packed pink staining plates of hepatocytes. So the outer layer is in a form of collagenous tissue. We call it as a Gleason's capsule. And most of the collagenous connective tissue in liver is found in the portal tract. And also it have the bile duct and different types hepatic venule that drain the liver, means remove the bloods from the liver. And this is the pancreas. Pancreas is a lobulated gland covered by thin collagenous capsule which extend as delicate septa. And also there is an interlobular ducts, and islet of Langerhans is present here, and there is a adipocytes are scattered throughout the parenchyma. So this is the adipocytes. And this is the adenocarcinoma of pancreas, pancreatic adenocarcinoma, usually arise in the head of the gland, and T is a tumor with normal pancreas in the upper field. So this is the normal. This is already uncontrollable proliferating cells. So tumor develop here. And there is a irregular glands, the G with collagenous stroma, and normal pancreatic acina cell is here. So next is urinary system. So this is a kidney shape. It has a cup shaped calyx surrounding the renal papilla here, and it has the regions of the cortex with the scattered or numerous renal corpuscle. So this is the endocrine system, made up our different types of glands. So it have the anterior pituitary and also the posterior pituitary. And this is a thyroid gland, contain numerous thyroid follicles. Okay, so this is part of the central nervous system, the brain meninges, covering the membrane and our spinal cord. So it have fibrous strands. Here, can you see transversing the subarachnoid space of the brain meninges. So there are small vessel. Here can be seen small vessel. Okay, so and some penetrating vessel also seen by perivascular space. So this is the brain meninges covering our organs of CNS. And also the last one, special sense organs. For example, the taste bud. So this is a micrograph of the taste bud, and this is the P is actually the taste pore, the opening at the surface. And olfactory, sense of smell. So located, there is a terminal bar here at the luminal surface, lining with the epithelium. And the eyes, there are 3 layers of the wall of the eyes. So the pigment epithelium, the choroid, the sclera, and also the retina. So different layer of wall of the eyes. And also the 10 distinct layer of the retina. So the main types of cell is, which is the neuron, pigmented epithelial cells, and also neuron support cell. There are 3 cell types, different cells working together that made up our retina. So that's all for the overview or examples of the tissue organization in our organ system.